Hello guys. Okay, in this video we are going to share about glycolysis, Krebs cycle and electron transport chain. Okay, now let's go.
Glycolysis. Glycolysis is the metabolic pathway that converts glucose into pyruvate. After glycolysis, there are three possible fates for pyruvate. Firstly, anaerobic glycolysis. Secondly, aerobic oxidation. And thirdly, anaerobic fermentation. There are consist 6 steps in this glycolysis metabolic pathway. There are two phases in glycolysis. Phase 1, preparative phase, which is 6 carbon sugar convert into 3 carbon sugar. Phase 2, ATP generating phase, which is produce energy in the form of ATPs and NADH. The overall reaction of glycolysis is glucose plus 2 NAD ion plus 2 ADP plus 2 Pi, which is produced 2 pyruvate plus 2 NADH plus 2 ATP plus 2 molecule of water plus 2 hydrogen ion. There is a net gain of 2 ATP per glucose molecule. As glucose oxidizes, 2 NAD ion are reduced to 2 NADH. The fate of NADH and pyruvate. NADH and pyruvate are energy, which is two possible fates. Firstly, aerobic. Oxygen is available. Pyruvates enter the citric acid cycle. NADH is reoxidized in the electron transport pathway, making ATP in oxidative phosphorylation. Secondly is anaerobic condition. NADH is reoxidized to NAD ion, providing additional NADH for more glycolysis. Pyruvate to lactate, which exists in muscle, and then pyruvate to ethanol, which consists in yeast. This is the diagram for the fate of NADH and pyruvate. Anaerobic conditions. There are two examples that I insert in this explanation. Firstly, lactic acid fermentation. This is the diagram about the glycolysis of lactic acid fermentation. Secondly is alcoholic fermentation. Also, I include the diagram of glycolysis process about alcoholic fermentation. Aerobic respiration. When oxygen is present, most organisms will undergo two more steps, which is Krebs cycle and electron transport, to produce their ATP. In eukaryotes, these processes occur in the mitochondria, while in prokaryotes, they occur in the cytoplasm.
Hello and welcome. In this video, we are going to talk about Krebs cycle. Krebs cycle is also known as tricarboxylic acid cycle, and it is a biochemical pathway that is used to generate energy through oxidation of acetyl-CoA. It is also used for the synthesis of NADH and for production of amino acid. Krebs cycle can take place in mitochondria of eukaryote or cytosol of prokaryote. The first step involved is actually a preparatory step, which begins with pyruvate. Pyruvate is derived through the glycolysis of glucose, and this split into two molecules of pyruvate. The next step is the oxidation of pyruvate into acetyl-CoA via the enzyme pyruvate 3-hydrogenase. In this reaction, a molecule of CO2, carbon dioxide, and NADH is generated. Acetyl-CoA will combine with oxaloacetate to form citrate. This reaction catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme BIOCHEMISTRY. Next, the isocitrate is oxidized into alpha ketoglutarate by the enzyme isocitrate dehydrogenase. In this reaction, a molecule of NAD is reduced into NADH, and a molecule of CO2, carbon dioxide, is generated. Next, the alpha ketoglutarate is converted into succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase. In this reaction, a molecule of NAD is reduced into NADH, and a molecule of CO2, carbon dioxide, is released. Pretty much the same from the previous step. For the next step, the succinyl-CoA is converted into succinate via the enzyme succinyl-CoA synthetase. In this reaction, a molecule of guanine triphosphate (GTP) is generated. Next, succinate is converted into fumarate via the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2 is generated, which is used for production of FADH2. Next, the fumarate is then converted into malate via the enzyme fumarase. And last, the malate is converted into oxaloacetate by the enzyme of malate dehydrogenase. In this reaction too, NAD is reduced into NADH. Now, let's see the overall result of Krebs cycle. Through each cycle of the Krebs cycle, following product is generated: 3 molecule of NADH, 1 molecule of FADH2, 1 GTP, and 2 molecule of carbon dioxide. Now, since glucose is split into 2 pyruvate compounds, if you watch the glycolysis video, for each molecule of glucose, this cycle runs twice. Hence, the overall product formed is actually 6 NADH, 2 molecule of FADH2, 2 GTP, and 4 molecules of carbon dioxide. All of the product in Krebs cycle will next be fed into electron transport chain for the generation of ATP. And that's all the short explanation of Krebs cycle from me. Thank you very much.
NADH and FADH2 is the product of Krebs cycle. But do you know what is their function? For your information, NADH and FADH2 function as an electron carrier in electron transport system. Yes, electron transport system is a set of enzymes which is located on the inner mitochondrial membrane, and it is the main source of energy in the anaerobic pathway generation. NADH and FADH2 are used to produce ATP in a structure called cytochrome. Energy released by the electron captured as ATP by ADP molecule. The ADP then reduced to ATP by the process called oxidative phosphorylation. What is oxidative phosphorylation? Oxidative phosphorylation is the gradient exchange of H+ ion across the inner mitochondrial membrane. Now, let's proceed to the electron transport chain process. The process in the electron transport system begins when NADH and FADH2 enter the ETS, yielding 3 ATP per NADH molecule. Meanwhile, FADH2 enter at the coenzyme Q, yielding only 2 ATP per FADH2. NADH and FADH2 carry electron and proton to the electron transport chain located in the membrane. Energy from the transfer of electron along the chain transport proton across the membrane and creates an electrochemical gradient. As the accumulating proton follow the electrochemical gradient back across the membrane through an ATP synthase complex, the movement of the proton provide energy synthesizing ATP from ADP and phosphate. At the end of ETS, 2 proton, 2 electron, and half of an oxygen molecule combine to form water. Since oxygen is the final electron acceptor, the process is called aerobic respiration.